Species diversity is a combination of both species richness and abundance. It could be measured by various mathematical expressions named as diversity indices. To compare the diversity of different communities or ecosystems, you will need to collect data from at least two different plant or animal communities. When we are comparing ecosystems using these diversity indices, those ecosystems need to have equal sample size or sample areas. When we studying the whole diversity of particular ecosystem, it is difficult to count all the number of species at that habitat at once. Therefore, we place a series of squares of a set size or quadrant at the habitat and the species within the squares are identified and recorded. This technique is called as quadrant sampling technique. As a practical demonstration, we are going to place a line transect about 20 meters. To identify the transect, flags are placed in 5-meter intervals along the transect. We toss a coin to decide the placement site for the random quadrant sampling. After we throw the quadrant to the selected site. Then identify each species present in the quadrant and count the number of individuals in each species. When you come across a species that you cannot identify, classify it as unknown species. It is important to label those collected unknown species to prevent from using the same label for different unknowns. Continue this process until you won't get any new species. To assume that we have identified and recorded all the species at that area, we have to plot a graph of quadrant versus cumulative number of species, which shows that the cumulative number of species curve has reached into a static status. For study the diversity of a habitat, we have many diversity indices such as Shannon-Wiener diversity index, Markev's diversity index, and Simpson diversity index.